I'd like to welcome everyone to the Survivors Inspire Peer Network meeting this evening. Our special guest speaker is Barbara Van Heer. And Barbara, I'm so glad you're with us. Barbara and I go way back to 2017 when we first started to collaborate on what was then called the Cancer Cell Line, a pilot project to bring rare cancers together for the sake of genomic profiling and screening. And it has now advanced to the Cancer Cell Line Factory, and then now the Rare Cancer Dependency Map project. And with Barbara and Dr. Jesse Bohm and Dr. Dimitri at the helm of this, with a patient advocacy team. And we're just real happy that this has been such a successful initiative, Barbara. And we appreciate you being here to really explain to us how important this is to patients and the fact that it is a patient-driven program and what it means for global research in the future. Thank you.
Thank you for having me, Annie. As mentioned, I'm Barbara Van Heer, and I am with the Rare Cancer Research Foundation and Pattern.org. And I am going to share my screen. Am I coming up?
Yes.
Everyone see this?
Mm-hmm.
Okay, great. So I'm just going to spend a few minutes to talk to you about tissue donation and Pattern.org and how we're working with the LMS community to help further research. So tissue donation, a lot of people don't totally understand what's so important about tissue donation. And it's really the cornerstone for research. It's really critical for basic research in that once there's fresh tissue, scientists and even stored tissue can better understand the biology of the disease. And as we're learning more year by year, each disease is different, and even within the same disease, there are a lot of differences. And so having access to patient tissue really is helping to uncover all the differences there are between and within diseases. It can also help identify ways of new diagnosis and prevention methods, and also help identify potential new therapies and cures. About 4.5 years ago, the Rare Cancer Research Foundation launched an initiative called Pattern.org. And the real reason it was launched was to enable cancer patients to be able to direct tissue from their tumors, as well as medical data, to high impact research at leading labs. For those that may not be totally familiar with the research landscape and different things that have occurred in the past years, is that it's fairly typical for institutions. If a patient is having surgery at a particular institution, usually that tissue doesn't leave that institution. And since most rare cancers, by the fact that they're rare, it's not common for any single institution to be able to see a lot of patients in any given year. Some may see more than others, but overall, it's just hard. Tissue is hard to come by. And so as a result, less common cancers like LMS aren't represented in key studies or in clinical trials. And also researchers don't have access to that tissue. And as a result of researchers not having access to the tissue, not only do they not get research accomplished as fast as they could, but also there may be a lack of researchers in any given field for a rare cancer because they haven't had those tissues to study. And so Pattern.org was born so that it was basically a direct from patient approach where patients could go onto Pattern.org, learn about different studies or a study that is studying their disease, read the consent on what it means to be a part of that study. And should they choose, they can consent directly online to provide excess tissue from their upcoming surgery or potentially from a fluid drain for that tissue to go to research. That way, regardless of where a patient is being seen anywhere in the continental US, whether it be an academic center or whether it's a local regional or even just in a small town hospital. They can contribute to research where maybe they wouldn't have had the opportunity to do that in the past. So basically, Pattern enables patients to be put at the center and determine where they want their tissue to go to get it to leading labs where new therapies and learnings can occur. And how people find out, how patients find out about Pattern is that we work directly with quite a large number of foundations, such as LMSF, so that they can let their patient communities know about the opportunity to contribute to a research project, should they so choose. So how it works, if a patient has a surgery coming up in the next 6 to 12 months, they can go to pattern.org, learn about a study that might be interested in their particular disease. They can consent online, and then once they consent online, we literally are notified within minutes of their consent. And then we start coordinating that tissue contribution by reaching out to the institution where the surgery is going to take place. We work, it varies by each case because each case is handled individually. We'll coordinate with the surgeon, maybe the oncologist, maybe the pathologist, maybe the operating room staff. Again, each case may be different. And we send in a kit, which I'll show you here, that has virtually everything included in it that that hospital will need in order to provide it's a tiny piece of tissue is all we need. And then if there's blood available, we would like to get blood and or normal adjacent tissue, all which would be collected in the normal process of a surgery. No tissue is taken that is needed for clinical care. So if it's a small tumor and there's not very much tissue, of course, the first priority is for whatever that hospital needs to help determine the correct path after surgery is what comes first. We're only looking for excess tissue. But we try to make it really easy for the doctor to participate in this. And we also try to make it really easy for the patient because we know that if they're preparing for surgery, they have a lot on their mind. So really, once they consent, there's nothing else they need to do. We'll take it from there. The only other thing that we ask is we will send them a medical release form from the institution where they're having surgery because most medical record departments won't accept an electronic consent. So we can't do that at the same time as the tissue donation consent. And we would send that to the patient, again, already prepaid so that they can send it back to us with ease. To date, we have collected from, this is just a sampling of the hospitals where we've had patients consent. And the majority of those, we've been able to collect the tissue. And specifically for LMS, these are all the institutions in the US where the project with the Broad Institute that Annie referenced, where patients have consented. To date, we've had 37 patients, LMS patients, consent to the project across 28 different institutions. Of those 37 patients, we've actually been able to collect 20 fresh tissue samples from 20 different institutions. And you may wonder, well, but 37 patients consented and you only got 20, what happened to the other 17? Many times we have patients that consent and for one reason or another, their surgery doesn't occur. It may be the physician decides that they're going to go on treatment first and they may not need a surgery. Their surgery is canceled for some other reason. There are lots of different reasons, but we've had really strong success and support from the patient community with LMS, as well as from the various surgeons that we've worked with all across the nation. And so I want to say thank you if any of you have participated in the past or if you know anyone that has. And I'll tell you shortly what happens to that tissue currently when we receive it. So the projects that we're associated with, to date, we've really focused on living tissue, which means tissue that hasn't been stored in a biobank. It's literally directly post-surgery. It's not put in a preservative. It is put into a little tube where it has a media condition so it can continue living until it gets to where it's going to go and they do with it what they need to do to keep it living. But that tissue is used in research for what are called basically living cancer models. They're called cell lines and organoids, and a PDX model basically means a patient-derived xenograft, which would be a sample of that little mouse. A cell line is basically, and I'm not a scientist, so I apologize if any scientists are listening, but it is a growth of those tissues that's not a three-dimensional growth, but it kind of grows across and is rather flat. And the goal would be to create cell lines that are ongoing in life, that replicate the disease similar to what it would be in the human body. And an organoid is basically the equivalent of a 3D cell line. Also, with these cancer models in the tissue, labs can generate robust genomic data, as well as potentially do drug screening to see what's really causing, what is that cancer dependent on to live, and what maybe could the researchers change about that so that that cancer no longer survives. So as Annie mentioned, we've been working closely with Annie and Mitch and the Broad Institute. Originally, the project was the Cancer Cell Line Factory, which is where they're taking an innovative approach to cell line model derivation. One of the fascinating things about living models is, particularly for rare cancers, most of them are very hard to create. And once they are created, sometimes they don't last very long. They may live for a while and then not anymore, or diseases that have had what are called these living models. Researchers may have had them stored in biobanks, and they don't get to see other researchers, or researchers don't have access to them. And in fact, in some cases, maybe they're not really the disease they're purported to be. And so one of the very exciting things about the Broad is once they get the tissue, they have about 64 different media conditions. They put the tissue in to see what's going to make it have the best chance at growth. And they will watch and see which of those conditions, or which multitude of conditions, allow for that cell line to generate. And once they are successful in making a cell line, I'm very excited to say that as the project went to kind of the second phase, the first phase was to create the models, and there are still tissues there that are in the process of growing, and they're watching those, hoping to create long-term models. But a number of models have already been made, and 3 of those models that have been made are actually headed to kind of the phase 2 of the project, which is called the Dependency Map project. And literally the purpose of that is to see what is allowing that cancer to live, and what can they do to change about that particular cancer model to see what could be changed about it so that it doesn't live, or it can be controlled by drugs. And so within the dependency map, they'll be doing CRISPR screening as well as drug screening. And an even more exciting part about this particular project versus a lot of projects that are done in research is everything that comes out of this work. So the models, any of the genomic information that's created, or any of the learnings from the dependency map portion will all be placed in the public domain. So what that means is all of that data and those models aren't going to stay in a particular researcher's lab. it's going to be, the models will be put in a biorepository, a public biorepository, where researchers all over the world will have access, and they can actually order those models to be sent to their labs where they can do more research. And the data also will be put in the public domain through a resource that the Broad Institute of MIT and Harvard has, and they release data to the public every 3 months. So the goal of trying to get these very critical research resources for specific diseases like LMS, the Broad is bringing to life in real time. Some of the key points in terms of if you are ever aware of a patient that might be interested or want to spread the word, is we really at pattern.org utilize the fresh tissue. So again, that's if someone's having surgery. Also, there are some diseases where ascites, which is a fluid buildup in the abdomen, or fluid buildup around the lungs, it's called pleural effusion, drains can occur. And in many cases, there are cancer cells included in that fluid. So we can collect from those. Any tissue that we would take would not be anything that is needed for the patient's care. It would not affect the patient's clinical care in any way. If the patient decides to participate in this research or doesn't, it doesn't affect the surgeon or the hospital. Everything goes just however the patient wants it to go. The sample size, we're asked this a lot, is how much is needed. The Broad would prefer 0.5 centimeters cubed, but can deal with less if need be. And if possible, we'll collect blood and adjacent normal tissue if possible. Because we're dealing with fresh tissue, we do need patients to consent prior. We would love to have a week's notice, but quite frankly, if a patient consents a day or two in advance, if there's any way we can make it possible to happen so that they can be a part of this research, we will do our best. Sometimes we have challenges because we have to reach out to the hospital and get them to agree to release the tissue, and we have to overnight a kit to them, et cetera, et cetera. So there are times when we can't make it happen, but we'll do our best to. For the Broad project, at this point, it is for general research only, which basically means is we can let the patient know that the tissue sample arrived at the Broad, but there's no other information we can release back to the specific patient. We can let like the foundation, like Annie and Mitch know, at any given point, there's so many samples we received, but we can't tell them anything about the patients that consented, all because of privacy regulations. So this is for general research only. A goal of this project, and actually any future projects that Pattern would be involved with, we would love to be able to give information back to the patient to help inform clinical decisions. We're not there yet, but trust me that that is definitely a goal that we have as well as our research partners. We protect patient privacy. The Broad Institute is not given any identifying information about the patient. Everything is de-identified that they receive, meaning they don't receive the patient's name or the patient's birth date. They receive information such as how old the patient is, if they're male or female, et cetera, et cetera. Any data that we collect is encrypted on a HIPAA compliant server. A patient can revoke consent at any time. There's no cost for a patient to participate. And the Broad is very interested in collecting more samples if anybody currently or down the road is interested. And kind of in a nutshell is our whole goal is to help rare cancer groups and communities put patients at the center, because it's because of patients that we do what we do. And we work with labs that have patients at the center of their research. And so we are glad to talk to anybody that wants to talk to us. If a patient has a question, they can contact me directly. They can on our website. reach out to us either through a chat box or a phone number, or if they have questions about, specifically questions about the research at the Broad Institute, there's also contact information online at pattern.org of who they can reach out to then. So that's kind of a high-level overview of who we are, why patient-directed research is so important. Is really we're trying to give patients some decision rights on if they want to contribute to research, that they can do it outside of their own institution, should they choose to do that. I will let you know that we work with a lot of doctors and patients that want to contribute to more than one study. In fact, I was working on one this afternoon where the patient actually is involved in 3 different studies at 3 different hospitals. Well, 2 hospitals, and then also the Broad Institute. They wanted their tissue sent to the Broad. So we help coordinate, knowing that the patient has some other things that they want to participate in, and the patient has given the surgical institution their priorities and told us their priorities of what they want to have happen. And so we're glad to work with patients on that as well. So I've talked a lot. I guess I'd like to open it up for questions, and I will stop sharing my screen, at least for the time being.
Well, we do have some questions in the chat, and I'd encourage, as far as the recording goes, if anyone has more questions, enter them in the chat for the recording. The first one we have there for you, Barbara, is, there's a set of identified sarcoma centers, and personally, I don't remember anybody asking me, like, hey, there is some research going on that would you like to donate some live tissue before my surgery? So any idea why they don't ask, like, all of their LMS patients,
Yeah.
hey, would you like to donate tissue?
So that's a really excellent question. And kind of the long and short of it is, the way we work is patient by patient and through patient foundations and patient groups. If we were to go to a sarcoma center and say, hey, do you want to work with us? Then what that requires, and this is historically the way research has been done, is that you have to negotiate what's called a material transfer agreement. And it's over the intellectual property of what may happen with that tissue down the road. And those negotiations can take 6 months to years to negotiate. So the way we get, quite frankly, these institutions to release the tissue, and trust me, at times there are battles behind the scenes that we have with the institutional bodies there, where they say the tissue is theirs because the surgery is happening at their institution. We fully believe that any tissue that's needed for clinical care, yes, the institution should have first priority on that. But if a patient has excess tissue, which at many institutions, it will go in a biobank. And if they have researchers there that are going to do research with that tissue, that is incredible. We would fully support and say that is awesome. But unfortunately, in a lot of institutions, that tissue may never see the light of day. And in other institutions, that tissue may literally be tossed. So we were founded on the principle, we were founded by a rare cancer patient who was frustrated that research hadn't progressed faster, not only for his disease, but for other rare cancers. And so we are finding ways to kind of change the system to give patients more choice, to get these valuable tissues to researchers that are going to do great things with them, but also share, so that the way a lot of institutions get funding, and totally understand why it's been this way, is their researchers have to publish papers. And then those researchers are able to get grants and everything because they've published. The challenge with that is many of these researchers won't release their findings until after they're published, and publishing sometimes takes months, if not years. And so in the meantime, other researchers, if they knew what another researcher had found, maybe some other new advancements could come to place. So to be honest, we're kind of going against the status quo. And the way we're able to do that is because patients have basically enabled and requested that we get a little bit of the excess tissue to go to labs that will basically do good, not only for the patient community for the particular cancer, but in the case of the Broad, they'll take all the results they find from LMS and compare them against lots of other different rare and common cancers. And so there may be some commonalities they can find by sharing all of that data and sharing it broadly.
Right.
Kind
It's
of a
just tough to
long-winded-
get, it's just often tough to get to the patient before they have their surgery.
It is. It is. And,
So
and
we have another question, and it says, is this mainly for primary tumor sites, or does it also include metastasized mets?
either one. The researchers we're working with feel it's very important not only to try to solve the complexity of figuring out the primary tumor, but also those that have metastasized or may be resistant to treatment.
Okay. And what about patients that have biopsies prior to surgery? Might there be enough tissue or fluid there for analysis?
Basically, we are no longer, we've taken a few biopsy samples, and for a variety of reasons, including with discussions with the researchers, have decided not to accept biopsy samples. One is usually there's not enough tissue taken. Number two, the studies that we are supporting to date will only allow us to take excess tissue, and normally there's not excess tissue in a biopsy. So that's why we have to stick with a surgery. Now, at some point that might change. At the point where we're able to that the research labs that we have sent tissue to are able to potentially provide something back to the patient's clinical care team, we may have a whole different way in that, a patient could decide to contribute without it being excess tissue. But we're not there yet. So right now we can't take any biopsy tissue.
Okay, and what about a surgery that happens after chemotherapy? Is that tissue still okay to collect?
It is. It is.
Okay, and is there any age limits involved in the tissue samples?
There is not. The process is slightly different if a patient is under the age of 21. They'll still go to our site. Technically, they don't sign a consent form on our site. They will actually need to talk to a person and sign, because of the way the review boards that oversee the research. But they can still participate in the project. We just have a little extra step of having them talk to a person and sending them, we can do it electronically, a form for them to sign, and then they can mail it back. We can, send them a postage paid envelope to send it back to us.
Okay, and Annie, you have a few good questions there. Let me let you ask those directly.
Okay, I can't see what I wrote here.
About clarifying the CRISPR screening process and purpose
Yes.
in terms of the genomic characterization of the LMS tumor tissue.
Barbara, I know CRISPR was featured in 60 Minutes at one time. And
Yes.
really rolled out what that was.
So I'm not an expert since I'm not a scientist. So we would really need somebody like Jesse to fully be able to describe CRISPR. But what CRISPR, the way that I understand it, and I have a fairly simple brain when it comes to science, I will tell you, is that CRISPR can go in and see what's going on in your genomic profile of the tissue, and like cut out parts that are, and kind of move some things around to see, like, I guess an example is with sickle cell anemia. There is one mutation that causes that disease that's prevalent in the African American community. And the thought is with CRISPR, like they could cut out that the part that's mutated, in order so that in the future the patient wouldn't have that disease. And there are a lot of different other things that CRISPR can do, but I'll be honest, I am not an expert. So
And the screening of repurposed drugs.
yes, so part of the Broad project is where they will test thousands upon thousands of compounds on, so the 3 LMS models that are getting ready to go through this pipeline, where they'll go through this CRISPR screening and the drug screens. Those living models, thousands of different drugs will be tested against them and in different combinations to see what, by doing basically what they call, I think it's perturbing that model, what they can learn from that in order to determine what drugs or what other things like they could do with CRISPR to change that so that the cancer either slows down or doesn't survive. That's in pretty simplistic terms, but.
Mhm. And what about organoids? How is that moving forward with LMS?
So again, that's a better question from somebody that works in the lab. And different cancers, they have more success growing a cell line versus an organoid. And the way it's been explained to me, and I've seen an organoid in a lab, an organoid is 3-dimensional. So like a cell line, if you saw it in a little, I'm going to call it a petri dish, its cell line is growing, then it's kind of flat, but the organoid kind of comes up off, so it's 3-dimensional. And different researchers have different opinions on which is better. Like pancreatic cancer, it's fairly easy to grow organoids with that. Some other cancers, organoids aren't very successful. And I can't tell you why that is. A researcher that understands it better than I would need to. But I believe most of the LMS in the Broad lab are cell lines, but I can double check that and confirm that back to you.
Okay, well, I appreciate that. Our foundation is certainly so glad that we're able to fund these cell lines to go
Yes.
forward. It's very important that we fund that.
It is critical
Yeah.
for new learnings to come from these cell lines. Yeah. And again, these cell lines, besides going to the dependency map to learn what's really happening with that cancer, those models will also go to a public biorepository so that other labs also will have access to those.
Mm-hmm. And all that said, the collaboration that has crossed over to our International Research Roundtable for the work group of working on cell lines and PDX models that Dr. Hemming is heading up. Can you give us a little bit of tidbit on that as far as his discussion with Dr. Bohm?
Honestly, I can't because I haven't been totally in the loop on that. So I'm sorry, I don't have an update on that. Okay. I can ask Jesse when I talk to him next week, but I don't know. I'm sorry.
Okay. And in the collaboration with MD Anderson coming up, is that something you can speak to?
So I can speak a little to that. So as I mentioned, pattern.org has been working with the Broad Institute, and they're creating these cell lines for LMS and other rare cancers. Within the next month, we will also be doing a project with MD Anderson. Similar type project. There's going to be a lot of genomic work being done from the samples that are sent to them. They also are going to try to create models. And in kind of an ironic twist, they'll be trying to create models either at MD Anderson, or in some cases may send the samples up to the Broad Institute of MIT and Harvard to the same project we're supporting. So it's all good. Everybody's kind of working together.
That's great.
Longer term, one of the things we're working on that MD Anderson has asked us how to do it with them is they would like to take, they have the ability to access what's called a CLIA lab, which means a lab that's certified for clinical research, meaning they could take part of the tissue, do some work on it in the lab, and potentially find some results that could help inform clinical care, potentially drug treatments that might work for that patient, potentially clinical trials that might work for that patient, et cetera, et cetera. So we're in the early discussion phases with them. They have the capability of doing that, but there's lots we have to do at Pattern in terms of certification, et cetera, and making sure, because once you start giving individual results back to a clinical care team, there are just lots of regulations and good regulations that have to be put into place to be able to do that. So that is down the road. I don't know if that will be 12 months or 2 years, but that is going to be an additional part, we hope, of the project with MD Anderson.
That's exciting. That's really, really exciting. Thank you for explaining that.
Another question, if there's a tissue that where they had neoadjuvant radiation, is radiated tissue also accepted?
It is. It is. So because of whether you're doing chemo or radiation, some of that tissue may have survived those treatments. And so if they have, researchers need to find out what it is that's allowed that cancer to survive. And so that's why these researchers are also interested in treated samples.
Very interesting.
I think this is so important because LMS in particular doesn't have a lot of proteins or biomarkers. And so the gene mutation is limited, and that how will this potentially impact immunotherapy research down the road, Barbara?
Oh, gee, you're asking me scientific questions that I'm not sure I'm capable of answering. I think the more projects like the dependency map at the Broad, or the work MD Anderson's doing, and a lot of other research centers, the more they learn about the cancer and what's driving it for it to continue to live and survive, the more they'll know on potential effects immunotherapy could have on the cancer. And they're just, how should I say that? With so many rare cancers, they're learning now. I mean, if you just think of what was known 10 years ago versus 20 years ago, and what's going to be known next year versus this year, findings are being, discovered. I don't want to say daily, but new things are being learned. And I can't answer specifically because I'm not a scientist, but that would be a good question for Jesse.
Okay, thank you.
I think someone has asked if tissue samples from surgery several years ago can be used. For the projects we're involved with today, they can't, because most likely they are stored either they were treated with a formalin so that they're no longer living. They may be stored like in a block, but that doesn't mean there might not be future studies that those could be used for. But for the ones we're participating in today, we need it from fresh tissue.
So in that case, should somebody sign up for Broad, or I think the answer is not sign up now.
Not sign up now. For the Broad, for everything that pattern.org is involved with today, it is just living tissue, which means from at surgery time, basically at drain or surgery time.
Okay.
There's also stored tissue, which is in a lot of biobanks, can even be harder to get out of an institution, because once the tissue technically comes out of your body, there's not agreed upon law on who owns that tissue. The vast majority of institutions would say it is theirs. So sometimes it's particularly difficult to get stored tissue for them to release it.
So we should take it home in a jar, right?
Well, not exactly. Some institutions are getting better. Patients, I have to say, are being are enabling themselves. The more that they know in terms of what to ask and how to ask it, is allowing more institutions to see there's really, the patients have power. I mean, they're the ones that, quite frankly, grew these tumors, and should have some rights to them in terms of if they want them to go somewhere, they should be allowed to do that. At least that's my perspective. And I know a number of patients I've talked to, one several years ago actually had a brain tumor, and her institution said they wouldn't release any tissue outside of where she was having surgery. And she kind of had a stand down with this major academic center, and they ended up releasing the tissue. She wanted it for a different project, but they released it. She said she should be able to take it home and put it in her garage, which she didn't want to do, and she ended up not doing, but it made the point, and they did release it. So I have seen in the past few years, the institutions are allowing the patients to have a lot more say, which is really, in my opinion, very encouraging.
Very good.
Yeah, that is.
I've got a question.
There you go.
Can I speak?
Yes.
This is Denny.
Oh.
Yeah, I'm a patient at MD Anderson with UPS, and I was glad to on Jeff's questions were excellent, and I was wondering, because everything I'm like 48 months of no evidence of disease, so all of my surgeries and everything for the time being are over with, so I'm just kind of in a surveillance mode. But you know, everything, and I'm on what they call the patient advisory family program volunteer at MD Anderson, and they're very patient centered. Not that they haven't been, but they're putting a lot of emphasis on patient experience. And I was a little bit, and there's probably legal reasons, but I was surprised that they don't promote or encourage the patients there to, you know, submit their or authorize or approve that, because I never hear that when I'm up at the center, you know, about do you explain to them about the program. I think a lot of people would participate.
And MD Anderson, quite frankly, has been wonderful for us to work with. And even before we started collaborating with them, they were very good when patients did know about the project with whatever rare cancer they had. In working with them, they would release the tissue to us. I can't say that for all institutions, but they were really pretty incredible. Historically, the academic institutions have studies going on in their own labs or in collaborations with other hospitals. So they kind of take priority. And so I think they're doing really good research. And as I say, not all institutions necessarily operate that same way, but I've been very pleased to see what they have done with us and working with us as a partner, and before we were even a partner, in terms of releasing patient tissues. As I say, we're just kind of the middleman. We don't determine which research is better. The tissue never actually comes to us. We kind of handle the consent and the biologistics, and, you know, help let patient groups know about what we do and work with the laboratories. So we just kind of try to get everybody to work together. That's kind of our role.
Great. Well, I've talked to Annie a little bit, and if there's anything I can do to help Annie, I will, you know, up at MD Anderson.
Thank you.
Appreciate that. And
MD Anderson is working closely with the Broad on rare cancers, and they have a separate agreement that some of the rare cancer samples they get at MD Anderson from patients being treated there, they'll actually send up to the Broad for the Broad to do work. So in the case of the Broad and us and MD Anderson, we're all working together, some in kind of a different projects, but they all go towards the same goal, I guess I would say.
I understand.
And even in the COVID climate now, you're still accepting tissue. And I know when we've come in the past 2 years to be with you at the meetings, you've said that LMS was one of the leading
It is.
responders of this program. And is it still the case?
It is. So every group is different. Every foundation works differently. Each cancer patient group is different. And I have to say the LMS community and Annie and Mitch have all been absolutely not only a joy to work with, but understood the priority and have generously donated tissues. It's absolutely one of the top groups we've had in terms of patient participation in agreeing to allow their tissue to go to the Broad. And so I can say on behalf of pattern.org, thank you to the patient community for supporting research and hoping that this research will lead to some great things so that at some point down the road, every LMS and other rare cancer survivor is a long-term survivor, and that we, even if we don't come up with a cure, but that it's a treatable disease so that it doesn't affect lifespan. That would be our ultimate goal, to help as many of these groups as possible. And you guys have just been terrific. I wish every rare cancer group that I worked with was as understanding and wonderful to work with as all of you and your other patient survivors have been.
Well, we really appreciate you and your mission, and we appreciate that the patients have been responsive, and the fact that moving forward together in this is so imperative to move treatment protocols and clinical trials forward faster is really a testament to the commitment of the research community, and for the patients to respond to helping them do that, because it is a patient-driven partnership. That really is key and important. So we appreciate that very much.
Hi, Andy. This is Mitch. Can I say
Yes,
something?
yes, absolutely.
Hi, Barbara. This is Mitch Ashe. I'm driving home from work, so I've been
Hi,
listening to you. But I wanted to say a couple things. One was how tremendous your program is and how helpful it's going to be going forward for the development of advanced research. Andy had asked the question about our international roundtable, and one of the issues was we need to go back to the basic science, which you alluded to earlier. And the only way we can do that is to have models to use to do that type of research. And that's why we were so enthused when we were able to apply and get accepted by the Broad to develop these, because there are a lot of people that say they have cell lines, but they're really, as you analyze them, are not pure cell lines. And we know that those coming out of the Broad will be available to the entire international community to do research if they choose to, plus to use the data that they're going to accumulate as they analyze these. And the other thing that I think is important about your program, it's good that we have the big research centers and the big sarcoma centers. Unfortunately, as you well know, many of the patients have had surgery before they got there. And I think one of the strengths of your program is that you have worked with many, many, many community hospitals, because by the time the patient gets to the other center, that there is no fresh frozen tissue to be sent. And I think that's a real strong aspect of your program, is that you can contact any small hospital anywhere in the country, and those patients can participate, which I think is great.
Absolutely. Yeah, we'll work with anyone. In most of the smaller hospitals, it's rather fascinating. Most of them have never been asked to share someone's tissue, and like the doctors are glad to share it, but they don't know if they can because nobody's ever asked them. Like a lot of these hospitals, they don't store it. They usually just toss it. I mean, they keep enough for whatever they need to do the testing on for the patient care, but then they just toss the rest. So they're like, well, yeah, we'd be glad to do it, but we don't even know who to ask to make sure we can do it. So but they've all been, all the hospitals, each one is unique, each case is unique, because you can work with the same hospital, but two different doctors and two different experiences in terms of, explaining the program and getting them to do it. But I'd say, vast majority of the folks that we work with at the hospitals have been extremely supportive. And we've even received notes back from some of the nurses we've worked with and the doctors on how they've never seen rare cancer tissue being utilized in this way. And they're just thrilled to see it, because before it either went into a black hole or it just wasn't going to enough places to support the work that needed to be done to try to help patients sooner versus later. So we appreciate your whole community. And if there are questions for me, feel free to contact me directly or go through Annie. I'm pretty accessible. We're involved with a lot of projects and things going on these days, but always glad to talk to any patient if there are questions or if they have ideas. Always glad to listen.
We thank you so much, Barbara, for your time. Thank you so much for this.
You are very welcome. Thank you for inviting me to talk to you all this evening.
Thank you.